And thank you. And yeah, hi everyone. My name is Carol Poole. I'm currently the Acting Director for Biodiversity Assessments at SANBI. And I'd like to welcome you all to this webinar. We are, I'm going to ask my colleague Divadine to explain a little bit about Reverse the Red and where we're coming from. But from this perspective, it's really great to have Reverse the Red here early in February because our National Biodiversity Assessment just came out in December last year. So we hope to connect the dots of species threat status and the recovery actions. We've got a very exciting lineup today, and I just want to also let you know that there is a second webinar next week, Friday, that will focus on the aquatic things, so freshwater species, marine species. So we will send information about that webinar as well. Make sure you watch out on the SANBI show socials and things like that. Please keep your cameras and microphones off and everything. You're welcome to introduce yourself in the chat, especially if you are an outsider not from SANBI. We did actually distribute the notice of this webinar quite widely, so we'd love to welcome anyone who's sort of from outside SANBI, and especially I see some of our French colleagues in the room as well, so welcome, welcome, welcome to all of you. So please do introduce yourselves in the chat. We won't have time to go around the room. We do have a full lineup, so we're going to get moving quite soon. We're going to talk, Divadine's going to get an overview so that all of you can understand where we're sitting at. We're going to then have mammals, plants, a particular case study on in the endangered Rose's dwarf mountain toadlet, so we're covering amphibians, and then we're going to have butterflies as well. So that's the lineup for today. If you have questions, I'm hoping that there will be a short few minutes or two after each speaker for questions. But just in case we don't, please pop your questions in the chat while we're going. I hope you can all access the chat. And then if we're running slightly behind, what we'll do is the speaker will answer the questions as soon as they finish speaking. They'll see the chat and try and answer your questions, and then hopefully we'll have a little bit of time at the end of the webinar to answer any remaining questions. All right, without further ado, I'm going to hand over to Divadine. Did I cover everything, Divadine? And over to you. Thank you.
Thanks, Carol. I think you did well there. Everything's covered. I'm just going to start sharing. Sorry about seeing the calendar. So trying to get that away. So as Carol was saying, I'll be presenting on the National Biodiversity Assessment species finding, and then connecting it to today's presentations about how we reverse the red on these threat statuses. Okay, so I hope most people in the room know what the NBA is, the National Biodiversity Assessment. It is conducted across all of the territories of South Africa, and today I'll be focusing on the terrestrial realm results for species. Esetu, if you can pop the link to the NBA website in the chat. So as I'm going, you can click through the NBA website as well if you've got a second screen, but please keep focus on the presentation. So the NBA is conducted on 3 levels: ecosystem, species, and genes. And as I mentioned, we're focusing on the species and specifically the threat status of species and the trends we've seen over time as reported in the 2025 NBA. An important acknowledgement is that this MBA is a collaborative effort. We have many partners, many contributors, and if your institution's logo is not here, please go to the website, and where you've contributed, that's where the acknowledgements are. So it's very important to know that all of this work is a culmination of many, many years of investment and time into being able to measure the state of biodiversity in South Africa. Because of all this work, South Africa is a world leader in being able to assess the state of our biodiversity. And as a result of that, in collaboration with our French colleagues, as Carol mentioned earlier, we've developed mapping biodiversity priorities, and also an e-learning course where other countries can learn how to also conduct the state of their biodiversity in their respective countries. So please do share this information. So back to the species. Our species work is not possible without our species experts. So in the next slide, I'll speak to the National Species Expert Group of South Africa. Then our provincial agencies, these are the agencies that are doing the conservation work on the ground, being able to monitor and apply the expertise and also feeding back to SANBI when we need information on some of other indicators that we measure. The NGOs that we collaborate with, some of them also conducting assessments, providing data, so their role is immensely important, especially with limited capacity within government organizations. And then the role of ordinary South Africans, our volunteers that take part in citizen science projects, some you'll hear about in the presentations to come, showing how important it is for us to know what's happening on the ground so that can inform the assessments that we do, and then in return also feedback into protecting those species better. So our National South African Species Specialist Group, in the last 2 years, we've become an official IUCN expert group, but those of you that have worked with SANBI over the years know that this expert base has developed over more than 15 years, and South African species experts, the conservation agencies, has contributed to decisions that are made and tools that are developed to support decision-making for species conservation and also to prevent further loss in species in South Africa. Myself and Linda Harris are the co-chairs of this group. And over the last 2 years, we've supported coordinating the experts to contribute to the NBA that has recently been published. And also, this is a source of information where the experts are on the ground. They're researching species and they know what is happening and are able to feed this back into the assessment process. So going into the assessment, I'll only be focusing on the status of species in this presentation because of limits of time, but over the next few months, you'll see some other talks coming up about the NBA findings, so please look out for them. So in the previous NBA, the 2018 one, we assessed 12 taxonomic groups. In this NBA, we've increased it to 16. A big increase has been in the invertebrate representation in this MBA, and that came out of 2018, where it was a finding that there's a gap in our understanding of invertebrate statuses in South Africa. So we've included freshwater invertebrates as well as terrestrial, and then also in the marine realm, expanded on the groups that we included in the assessment. You can find out all about these assessments in each of these taxonomic group pages on the NBA website. My colleague has posted the link in the chat so you're able to access it. And for all of the realms that we indicated, that I indicated earlier, there's a summary at the realm level. What is the state of species in the terrestrial realm, the marine realm, and the freshwater realm? So our key finding for species, and just to note, at the point of the 2050-2025 NBA, we have reassessed birds, mammals, amphibians, and reptiles, and those are the new assessments that you can see an end point here. We also have new assessments for Anastrocca, fairy shrimps, which is a new group included, freshwater crabs, and all of these groups, we use the IUCN Red List Index to look at trend over time. So how has species statuses changed over time due to impacts or due to improvements in their status due to actions and recovery work? In this graph, 1 means that no species are threatened, so our spiders are very high up. It's also a group that has many data deficient species, but also a large number of least concern species, about 2,200 species in that group. 0 meaning all species are extinct in the group, noting freshwater fish, low on this group, and the new group that we've included, sharks, the lowest Red List Index as one of our main findings. Overall, there's still a very negative trend for our species, with specifically mammals, plants, and reptiles showing decline due to climate change impacts, so extended drought periods, longer heat days, loss of habitat to mining and renewable energy developments. And these maps at the bottom here specifically indicates species that have increased in their threat status. So this map here is for plants. You can see most of the changes has happened in the Northern Cape on the West Coast, strongly linked to poaching activities as well as renewable energy and mining activities. While for animals, there's less of a structured pattern, but if you look at the individual taxonomic groups, there's more detail that can be unpacked. So this trend of species becoming more threatened leads us to respond to the biodiversity framework, where now we've assessed the status and we know that our species are becoming more threatened, so there needs to be action. South Africa, as a signatory to the Convention on Biological Diversity, we need to act to respond to Target 4, which states to halt species extinctions. And also, one of our findings in this MBA is to be able to respond to Target 4, we need to reverse the red, start recovery projects and enact recovery for those species that are on the brink of extinction and also species that are becoming more threatened. So over the last 2 years, within the specialists or the specialists expert group, we've asked our experts to identify which are the species in urgent need of recovery, that without any intervention, they will be heading to extinction in the next few years. So for these taxonomic groups, our experts identified those species. We've also asked them to estimate the cost of recovery of these species based on the past experience. And we have these preliminary lists and results of how much it will cost South Africa in the next 5 years. More recently, we've also engaged with the IUCN group that's identifying, they call it Target 4 species, and they've developed a more systematic process in identifying these species. And currently, our experts are comparing the results of the IUCN's process, where they use the red list criteria and categories to identify those species, to what our experts have identified. And I'm very happy to say there's strong alignment between what our experts have identified and what's coming out of the systematic process. So over the next few months, we'll be refining this list of species needing recovery. And for some of those species, we'll conduct a green status assessment, which is opposite of the red list. And it is there to track how well are we doing with recovery. So this is I'm just telling you about the reverse the red, why we're doing it. And the next presentations will show you some examples of how our experts have been involved with some recovery work that's predating the reverse the red, but will also contribute to our meeting our Target 4 target. Thank you, Carol.
Fantastic. Thanks, Divadine, for that overview. I'm going to ask Lazan to start sharing her screen and putting her presentation up. And we're going to move straight on to the next presenter. As I said, I might need to shivvy everyone along because of time. So next up is Lazan from the Endangered Wildlife Trust. They are a key partner on mammals in particular, but also several other species groups. Over to you, Lazan. And just a reminder to everyone, please, if you have any questions for Divadine, please pop them in the chat. We will try and get to questions at the end. Over to you, Lazan.
Thank you so much, Carol. And good afternoon, everyone. I'll be talking today about our mammal recovery projects in South Africa, and I'll just give you a broad overview following the latest revision of the Mammal Red List of South Africa, which we've just launched. So I'd like to start off with a couple of charismatic carnivores. The first one is the African wild dog, which is endangered on the National Red List. So why are we working on the recovery of a species? Well, that's because there are only around 120 adult dogs in South Africa. They are vulnerable to snaring and they are dependent on conservation. So what is being done? The Wild Dog Advisory Group, or WAG, acts as a national coordinating body linking government, researchers, NGOs, and reserve managers. Management of around 20 packs and 18 reserves are supported by WAG. There are also reintroductions of wild dogs into other African countries to expand the safe space available for wild dogs. And there's mitigation of conflict and engagement with communities to reduce livestock losses and threats like snaring. Who are the organizations involved in this? It's the EWT in partnership with private and community landholders and state conservation agencies. Moving on to the cheetah. Cheetah are vulnerable on the National Red List. Why are we doing recovery work on the species? Again, because there are only, we estimate, around 656 individuals in South Africa. They are persecuted outside of protected areas and they're dependent on conservation. What is being done? Quite similar to the wild dogs. There's translocations and reintroductions happening to move cheetahs between reserves to rebuild or to start new populations. There's genetic management to ensure diverse gene flow by sourcing suitable individuals and coordinating national movements. Habitat suitability assessments are done to evaluate how suitable a reserve is before relocating cheetahs. And there is monitoring work using radio and GPS collaring to ensure that introductions are successful and the cheetahs are adapting to their new homes. The organizations involved, again, the EWT, in a multi-partner collaboration effort involving NGOs, government agencies, private reserves, and conservation groups. Our next species is small and elusive. It's the riverine rabbit, which is endangered on the National Red List. Why are we working on the species? Because it has a small population, it's vulnerable to habitat degradation, and to the impacts of climate change. The work that is being done includes habitat restoration along rivers to ensure that the species recovers. Populations are monitored and some new populations are discovered using camera traps, and ecological research is done on these populations. Large connected conservancies and private reserves are being developed so that rabbits can move freely in the landscape, and biodiversity stewardship agreements are signed with private landowners to promote sustainable land use and protection of habitats. The organizations involved, Cape Nature, private nature reserves, the Northern Cape DENC, researchers and universities in collaboration with the EWT. Moving on to a species that is often in the news, the black rhino is endangered on the National Red List. I'm sure you're very well aware why we need to work on this, the recovery of the species. Poaching remains a huge threat and the species is very dependent on conservation efforts. So what is being done? There's a black rhino range expansion project which relocates rhinos to safe habitats. And since 2003, the project has created 17 new populations in South Africa, covering more than 360,000 hectares and translocating 270 black rhinos. These rhinos are microchipped and anti-poaching efforts continue. There's also community engagement to create large conservation areas that benefit not only rhinos, but a suite of other species. The organizations involved are WWF in partnership with private and community landholders and state conservation agencies. The last threatened species that I would like to highlight is the leopard. This species is vulnerable on the National Red List. Why are we focusing recovery efforts on the species? Although they're widespread outside of protected areas, they occur at low densities and there are severe threats to these animals, such as snaring and persecution. What is being done? Camera trap surveys have been conducted across landscapes to understand populations and movements of leopards. Key habitats and wildlife corridors are being protected so that leopards can move freely and safely within the landscapes. Human-leopard conflict is being reduced through better livestock management and relocation of conflict animals, and rescue and rehab of injured or displaced leopards is ongoing. The organizations involved are the Cape Leopard Trust, Ingwe Research Program, the EWT, and other partners. The Cape Mountain zebra is the least concerned species on the National Red List, meaning it's not threatened. The reason I've included it here is that it is one of the most successful large mammal recovery initiatives in South Africa. The species nearly went extinct about 100 years ago, and if it had been assessed at that time, it would have been critically endangered. So what was or is being done for the species? Well, they started off by protecting the last remaining herds and then growing the population in existing reserves. Currently, there is a project to fix genetic problems through a genetic rescue project, and there is an ongoing expansion of habitat by creating new reserves or restoring habitat. The organizations involved are Sandparks, Cape Nature, WWF SANBI, Sunborner Wildlife Reserve, and many other private landowners and private reserves. We don't have time to go into any of the other great work that's being done on recovering mammals in South Africa, but I just wanted to highlight 3 other species. They're all vulnerable on the red list: the bontebok, the ground pangolin, and the black-footed cat. Please go and look them up. There's some great work being done by these organizations that I've listed here. And at this point, I'd like to emphasize that species recovery work requires collaboration from multiple partners and is a long-term commitment. And then lastly, Davidine asked to just give an idea of how you can get involved in some of these projects. One of the main things we really need to know where the public can get involved is where the wild things actually are. So you can download the iNaturalist app and submit sightings of mammals to iNaturalist, especially small mammals. We're lacking data on many of our small mammals. And then awareness is key to making changes and having an impact. Please subscribe to newsletters or follow environmental organizations on social media and share our stories widely. And then we all know that funding is critical for all conservation work. So you can either donate directly or buy from retailers who support conservation. Thank you for your time.
Thank you so much, Lazar. That was really, really excellent. Some very, very fascinating numbers. I'm sure people will go and explore. Please go and explore the EWT Mammal Red List website, and of course the SANBI summary of the Mammal Red List status on the NBA website. Right, thank you very much. I'm going to move straight on to Tristan from our CREW program, the Custodians of Rare and Endangered Wildflowers program. Tristan, please go ahead and share your screen. Tristan's going to be talking to us about plant recovery programs in South Africa. Many plants, and we're only giving him 10 minutes. Over to you, Tristan. 7 minutes, in fact, we said to everyone.
Thanks, Carol. Appreciate it. Yeah, just the general introduction. Yeah, so my name is Tristan Kruger. So I support the work done by Ishmael Ibrahim here at the Custodians of Rare and Endangered Wildflowers based in Cape Town. And really just going to get straight into it. So as Davidine has so meticulously explained this, so most of our work is motivated by Target 4 within the GBF targets, because South Africa is a signatory to the Convention on Biological Diversity. And yeah, what this has been the entire motivation for all of our work, and how we've been doing that is actually going through the CREW data that has existed CREW has existed for more than 20 years, and with that data, we've been able to actually identify some of the species that are most in need of some sort of intervention. Otherwise, they're just going to fall off the edge of the world. Anyway, but how we've done that is using the CREW data over the last 20 odd years, and using a prioritization process, using the IUCN criteria, and then using various combinations of these, then creating a script and running it through the Red List database. And this first iteration of this list was produced in 2022, with 262 species produced. And then with the Red List update in 2024, that number went slightly up to 293. However, this is a crazy amount of plants to work on. Realistically, it's just impossible. So Ishmael and myself had to develop a decision tree, which helps us further refine the suitability of a species for species recovery. How this works is basically each species is run through the decision tree, and wherever it jumps out, it would be placed into one of 5 baskets, as we term them. And these baskets have their assigned conservation interventions which you will see in a minute. But really the goal is for species to make it to the end of this decision tree where they are considered a suitable recovery candidate. It's just a visual representation of this of these 5 baskets, and Ishmael has presented on this before, but so the monitoring required basket on the far left, that pertains a lot to the work that CREW is involved with. So actually updating the data that is available on these populations in the wild, with the other baskets either allocated to the Millennium Seed Bank Partnership or the gardens themselves. And then we've also been working on the suitable candidates for recovery. How we've been doing this is actually putting together recovery plans for these species, these suitable candidates. But I must highlight the fact that we've been trying to keep these recovery plans as brief as possible for the sake of the most important information actually standing out in these plans. Because, I mean, if you're filling the entire recovery plan with eco-geographical information and that forms the bulk, then, you know, it's just going to oversaturate things and you're not going to remember what's the important points in this recovery plan. Additionally, we've been in the process of putting together a review panel for these recovery plans so that they can be approved by not just us, but by our partners and various stakeholders, because at the end of the day, we all need to, we all bring our parts to play, and it just helps with a more holistic approach to the actual recovery process. Just an example of what's kind of in the recovery plan. So we've just got eco-geographical information, perhaps past and present projects going on, and the obvious objectives. Well, I haven't put all of them here, but this is just an example of the kinds of things that we will be putting in this recovery plan. Additionally, we've produced some monitoring material, so post-monitoring material. So once these plants have been planted out into the field, how do we go about actually monitoring them? And this is sort of a more professional approach to monitoring, because monitoring has been done in the past, but it hasn't been done meticulously to the point where we can actually understand what is our success rate. And I mean, yeah, at the end of the day, that's what we need to know. We need to know whether we are actually doing the job correctly, and what works and what doesn't work, so that we're not wasting our time and resources, which we all are lacking in. Another really cool little thing that we put together is the species recovery channel on the team's channel, species recovery for all of our conservation partners and SANBI staff involved. So this contains all of the products that I've mentioned before, all the lists, the decision tree itself. And then also we've produced this progress tracking spreadsheet, and this allows all of our stakeholders to actively update conservation actions taken for each of these 293 species. And we're just going to get on to some examples. So the first one I'll start with is just a quick update on the Marasmodes project. Again, Ishmael, this is one of his babies, but just an update since last year. But I'll just give a quick intro to what this genus is all about. So Marasmodes is a small daisy genus native to the Cape Lowlands and is highly threatened because of historical agricultural sprawl as well as urban sprawl. And as a result, it is most species, actually all species are threatened. And this particular one, Marasmodes undulata, is critically endangered and is limited to one site, 6-hectare site in Paarl. And Ishmael has been working for the last 8 years on recovering the species. Yeah, I'll just run you quickly through what's been happening. So yeah, you can just see a quick snapshot of the site itself. And what we've actually been doing is going out and planting these plants in winter so that they can establish themselves. And then for the first two years, we were not watering over the summer period with the expectation that some plants will survive and some plants will not. That would be a case of who's the strongest. And basically 3 plants have survived out of some of the plants that we've planted. But this has been very useful because it's actually allowing us to sort of identify where are the sites most suitable for these plants, and then actually focusing on planting more on the sites that have been successful. So just a quick recap. So of last year, of what was done last year. So as I mentioned, 3 of these plants survived the 2024-2025 summer. And then last winter, we planted out 148 plants in partnership with Stellenbosch University Botanical Garden staff and crew staff. And actually over this summer, there's been somewhat of an 80% survival rate, because instead of leaving the plants to just survive, we've gotten to the point where we've only got 7, well, it's always been the case, there's only 7 genetically distinct plants known. And so we can't really be bargaining with the little bit that we have. So we actually need to just make sure that we're watering these things through the summer so that we can get as many to survive through the summer and potentially survive longer and produce seed and so forth. And then nearly 400 plants have been produced between Crew and Stellenbosch University Botanical Gardens for the 2025 season. Another cool project that we've got going is we've been trying to get some signage going in the gardens just to highlight the plight of Maris modis, so that the public is aware of what's going on. It's a group of plants people are not aware of, and this is a great way of sort of getting out to the public and educating them about what's going on right on their doorstep with a little daisy that nobody knows about but is highly threatened and integral to the ecology of the lowlands. And additionally to this, we will also be planting out more Maris modis in the garden so that people can actually see what do these things look like. A really pretty one and a really cool project that we've got going in collaboration with Cape Nature and Stellenbosch University Botanical Gardens is the Diza barbata project. So this is a species also just limited to a little piece of sand fynbos outside of Atlantis, with less than 50 mature individuals. And basically what we've been doing over the last few years is just monitoring as well as hand pollinating some of these plants. So over the 2023 season, which was my first time that I got involved with this, we monitored the species alongside Cape Nature staff and Prof. Dirk Balstead from Stellenbosch University. However, all of these flowers that were observed were self-pollinated by the Cape Nature staff, and as a result of this, no seeding plants could be found. We assume that they are not self-fertile. And then over 2024 season, which was easily our most successful season, we opportunistically got out there and monitored the species as well as hand-pollinated all of the receptor flowers via cross-pollination. That's produced some beautiful, healthy seed capsules that you can see in the bottom. And these were collected and tissue cultured by our colleagues at Stellenbosch University Botanical Gardens. Last year, however, was not so great. So they were monitored and hand pollinated by, again, the Stellenbosch University Botanical Garden staff. However, poor seed set was the result is as a result of the very poor rains that we had. It was like the tap was just turned off, and unfortunately, no seed was produced for last year. And just a visual representation of kind of what it looks like. What we actually end up doing in the field is we're taking green pods, popping them in capsules, keeping them in humidity-controlled environments with silica gel, and eventually they make it onto some agar with a whole various combinations of various hormones and nutrients available in the middle, and then you get these little protocorms that form, and eventually you get something on the far right, which is actually little Diza barbatas that were planted out. And this is some photos sent to me by the late Donovan, who was very, very passionate, Donovan Kirkwood, sorry, who was very passionate about this work. Again, some more Donovan hands, but this is the last time I spent some time with Donovan in the field. But here we were actually baiting for the mycorrhizal fungi that is associated with Diza barbata. And the thing about Diza barbata, well, it is a terrestrial orchid, and terrestrial orchids have an association with a mycorrhizal fungi, and so they need to be in their seeds need to be in contact with this mycorrhizal fungi so that they can germinate. That's really just in a nutshell what is needed. So in order for us to actually capture the fungus itself, we actually need to seed bait them. and that involved putting seeds in the ground at the site, and at some stage through various methods we will be extracting the fungus itself. And then just another conservation action that we've been doing. So we did a hack last year, it was great, where the highest density of disers were. We went out and really got to it with taking out the Port Jacksons. And then just how can you get involved? So tomorrow, actually, the crew team and a few, Tiller will be joining us for a little bit of a hack down at Riverlands Nature Reserve where the diser barbatas are. And that will be followed by a bio-blitz at Riverlands Nature Reserve. And if you're interested, you can contact Ishmael Ibrahim, and there's his details. And I thank you very much.
Fantastic. Thanks, Tristan. You were about to get asked to stop, but you stopped just in time. Well done. This is really fascinating, guys. 20,000 and something plants in the country, 67% endemism. Yeah, I'm afraid we don't have time for questions yet. Please post your questions in the chat. We do need to keep moving on. And if we do have time towards the end, we're going to come back to more questions. Next up on the program is a recorded presentation by Professor Crystal Tolley from University of Johannesburg. Davidina, I'm going to share from my side. Is that okay? Or have you got it ready?
No, please do from your side. Thanks.
All right. Bear with me, everyone, because I've just got to share with my sound, and then you should hear the recorded presentation.
I am pleased to be able to introduce you to Rose's dwarf mountain toadlet, Copensi bufo rosei. This tiny little toad occurs only on the Cape Peninsula in an area of about 500 square kilometers in size inside of Table Mountain National Park. However, we've searched over the last 20 years throughout the distribution, and we can only find 2 active breeding sites. All the other previously known breeding sites appear to be locally extinct. There's no frogs there, there's no breeding, we can't find anything going on at those sites. The last time any frogs were recorded there was in the 1980s. So it means that the distribution size is more like 1.5 square kilometers, the total of these 2 areas, rather than 500. This results in a IUCN assessment of critically endangered due to the decline in the number of breeding sites or subpopulations, the number of individuals that were once at those sites, as well as the overall area. Of course, we want to know, where have they gone? Why have those sites disappeared? If they're in Table Mountain National Park, you would think that they have pristine habitat. So we've been monitoring since 2007 throughout the entire breeding season of those 2 active sites, as well as researching the extinct breeding sites. And what we have found through our monitoring is that the breeding is enhanced in open and disturbed sites. So that's like what you see here. It's a disturbed site, and there's lots of breeding going on in these tiny little puddles that have opened up in the fynbos. Where the fynbos grows up and becomes very dense, the breeding is excluded. They no longer will breed in sites like this. So anywhere where you have this situation, if they were previously breeding there, they will presumably go locally extinct, no more breeding. These open and disturbed sites are maintained by 2 processes that we can see. One is by grazing antelope that trampled down the vegetation, and that's why this veg here is trampled. It was the antelope, and in fact, some of these little puddles have formed in the footprints of these antelope, and the frogs are breeding right in the footprints. Unfortunately, the antelope are only present in a very small area of the park, so it's not a huge force. It is probably historically important, but at present, it is not a huge force for keeping the sites open. What is a bigger force, apparently, is fire. So whenever fire comes through one of our breeding sites, what we find is that that following year, the breeding is enhanced. There are many more puddles forming where the frogs come. There's thousands of frogs coming, and not just the breeding is enhanced, but the subsequent recruitment and survival is enhanced in the years after a fire. So these two forces are incredibly important for maintaining breeding, and the exclusion of these two forces may be what is causing the declines in some of those other areas. What we have been doing is trying to advocate for management burns to be at the site at regular intervals, and we can tell when this should be through our monitoring. When we start recording reduced breeding, we can advocate for a management burn to be carried out within the next few years. We'd also like to advocate for fire suppression to be at a minimum if the fire comes through at a site accidentally, but it's at an interval that's appropriate. We'd like to see it burn so that the site will open up. I should mention that the adults as well as metamorphs survive these fires. So we go up after a fire and we search the area for frogs, and we find plenty. We don't find mortality. What we find is live frogs there, and many, many metamorphs. So it seems as though the actual fire itself is not hugely detrimental, but what's detrimental is not having fire. Another possibility is to reestablish populations at the historical sites through introductions, but we really should only do that if those populations are truly locally extinct. And because this frog is so difficult to detect, it also has no advertisement call, so we can't just hear it and go toward a population to locate it. So we need to use other methods to find out if these populations are extinct or not. So we're using two methods. One is environmental DNA. We are ground truthing it at the two known sites. And if it can detect a signature of the frogs, we will then try the environmental DNA to see if it can pick up a signal at any of the extinct sites. The other method is detection dogs. The dogs have been trained at these two active breeding sites. They're very good at finding frogs. They can find adults, tadpoles, eggs.
Carol, you've muted yourself.
At these sites, we have to rethink our plan and perhaps rather manage the fynbos at those sites so the populations can grow naturally. And if we do reestablish the historical sites and do introductions there, we actually need to also know whether or not the environmental conditions are suitable. So they were last seen at these sites in the 1980s, and a lot of things, besides the vegetation, a lot of other things could have changed since that time. So we are measuring a load of environmental parameters at the two active breeding sites. And once we establish which, if any, we're measuring are important to the frogs, we will have to go to the extinct breeding sites and see if those sites still have the conditions that are good for these frogs. So that's where we are now with the interventions and hopefully recovery. And it's been a long road. We have been, as I mentioned, nearly 20 years on this. We've had a lot of support from institutions, but also researchers, interns, and students and volunteers over the years. And everyone has worked really hard to try to gather up the information we need to eventually reverse the red on this species.
There we go, everyone. Sorry about that. If you missed a piece of the presentation, I didn't mute myself. Someone else must have muted me. So hopefully you all heard that. It was just a couple of seconds that it was muted. But yeah, just a fascinating insight into one specific species. We are going to move straight on to the final presentation, and then hopefully we will have 5 or so minutes for questions afterwards. So there's Dave. Dave is with the Lepidopterist Society of South Africa, or Southern Africa, I think it is, and he's going to talk to us about Lepidoptera. Over to you, Dave. Can you share your screen? Oh dear, Dave has frozen a little bit. Are you there, Dave?
Yeah, I'm here. Okay. Can you see anything yet?
Not anything yet, Dave.
Oh. So Control Shift E. Should have something now.
Yes, it's coming. If you just can put it on slideshow mode, then we can see it. Yeah. There we go. Perfect. Over to you, Dave. Thanks.
Yeah, I'm going to first tell you about the COREL program. It was originated in 2011, and we started with 15 critically endangered species. And these are the steps you have to go through. Firstly, you've got to appoint a custodian. You find someone who's living nearby where this species is found to keep the travel costs down. And then they go and meet the landowner and introduce themselves and get the landowner excited about the butterfly by showing it to him. Always works. Then they start monitoring the butterfly population, going at the right times of year to do that, because butterflies only fly for a few weeks at a time. Then in the background, they search for other populations. That goes on for years and years and years. And then if they've got a good handle on the butterfly, then we try to get a researcher in there to find out what the ecology is and what the threats are. Then eventually, the final step is to formally protect the sites. So currently this program has grown, and we've now got 50 critical and endangered taxa being looked after by 16 custodians. And it's all funded by the Brenton Blue Foundation. It all started with the Brenton Blue. The Butterfly Reserve was up and going by 2000. Then there was a research program conducted by myself up until 2005. And it was discovered that the butterfly has a unique life history. The larvae actually feed not only on the leaves when they're very small, but then they go onto the rootstock. And so the life history of the plant is also important. So that gave us lots of food for thought. And it became a model for the COREL program. Because we said if we can repeat what we've done here, because basically we did recover the population to where it had been, and then we expanded the reserve from 1.5 to 12 hectares, and we were busy, making that suitable habitat. And then in 2017, a big fire came along, destroyed the butterfly reserve, and now we've spent the last 8 years searching for another population. In the little pictures, you'll see that there are, there's the butterflies mating, laying eggs, then there's the little caterpillars, then they go on the rootstock, then they go underground and are looked after by the ants. And then finally you get a butterfly if you're lucky. The ants themselves nest in dead wood, which was one of the reasons why they were very vulnerable to fire. And all the dead wood lying on the surface was all burnt up. So the butterflies haven't come back. Okay, the second one to look at, and it's got an interesting contrast with the other one. I chose 3 different butterflies that have got completely different life histories. This one, we had a type locality which had been known since the 1920s, which was still going strong. But then in 2013, fairly soon after we started searching, we found another site. So that was a big plus. We reduced the risk by 50%. We established a local management committee, and we've got a lot of really capable and excited people, botanists and people from the local conservation organizations, and the landowner himself. And he eventually, after seeing what all we were doing and witnessing this incredibly interesting butterfly, he agreed to have a conservation servitude on his farm. So that was the first one established for a butterfly in South Africa and showed us the way. The little pictures at the bottom are the life history. The one on your left would be the egg, which you can hardly see. It's a little black conical thing that sits on the rock. Then the larvae come out and start feeding on the lichen on the rock. You know, if you go in the field around rocks, you'll see that there's all these colored patches on the rocks. These are lichen, what these little caterpillars feed on. And then eventually they become a pupa, also a hairy little thing, and out pops some butterflies, the male on the left and the female on the right. You can see that the underside of the butterfly is quite different to the upper side. The underside is camouflage and makes it very difficult for any predators to spot it. And also it's quite cryptic when it's on the rock as well. Then there's Dixon's copper, Chryserites dixoni. It became extinct on the west coast in the 1990s, and lots of searching there. We've never been able to find it again. But miraculously, a second site was discovered in 1990 near Wetsand, which is the last place you'd expect to find it because that's on the south coast. How on earth does this rare butterfly have two sites that are so separate? But that's probably got to do with changes in sea level and all that kind of thing. The landowner, Stellenbosch University, committed to a 40-hectare contract nature reserve with Cape Nature. And that hasn't been signed and all the I's and T's crossed and I's dotted, but it's pretty much in the bag. The life history of this one is also very interesting. Everything goes on inside the ant's nest, which is the image on the right. These are what we call carton nests, and they're made by ants called cocktail ants, also known as chromatogastas to scientists. And if we look at the picture on the top left, that is actually a pair of butterflies mating, and they've just come out of the pupa, and they're sitting on the chromatogasta nest. And the first thing they do is mate. Now, from a survival point of view, this is very, very smart because the female then flies off and she will, there we go, from current slide. Yep, click, click, click. I don't know why we lost that. She immediately goes off and starts laying eggs. The third picture is actually the female laying eggs. And she usually lays them on dead material, on little twigs or even on rocks. And also quite close to where you've got scale insects. Scale insects, as the botanists will know, are insects that actually bore into the stems and suck out sap, and they, yeah. Anyway, the butterfly has several stages to its life history. It's got the ant that's looking after it inside the ant nest. And then this fourth picture on the bottom is actually shows you the honey gland, because on the back of the final instar larvae, and on the earlier instars, they've actually got this honey gland, which the ants are very, very keen to get the substance from there. Even when it's in the pupa, you can see the ants crawling over it, and they still pick up, the nutritious substance from there. This butterfly comes out of the ant nest, and then you can see the ant nest has got all these little holes in it, which are like a labyrinth of tunnels. And when they hatch out of the pupa, they crawl along these tunnels to the surface, and out they come. And if they're lucky, they meet their mate over here and they get started. So a fascinating life history this one has. And it's, yeah. So, okay, I'm going to give you a bit more stuff about how people can help. Well, I think the first thing you can do is to join the Lepidopterist Society of Africa, because conservation like this relies upon experts. And you can either join as an ordinary member and make a donation, or you can also become a sponsor and contribute more that way. And then if you're really interested in getting involved, you should approach me. You know, my email is readily available on various websites. And I can then introduce you to the local expert, and maybe you can go and see the butterfly yourself. And if you're really interested, learn plenty about it, and then eventually become, after about 5 years, an expert. So that's the way it works. So thank you very much for that. I'm going to stop sharing.
Thank you so much, Dave. That's brilliant.
Yeah.
Well done. Thank you. Great to hear from Lepsock again. Nice to see you again.
Yes, likewise, Carol. Yeah, you were my project manager.
Yes. All right, thanks everyone. We're making reasonable time and we have about 4 minutes left. So if anyone's got a burning question, I see Lizanne kindly answered 2 questions in the chat that were about genetic management, quite a big issue in translocations and things like that. So thanks for that. I saw someone had their hand up earlier. Jamie, would you have I think you put your hand up during Tristan's talk. Do you want to come in with your question? Was it a plant-based question? And if Jamie is still in the room. No, maybe not. Any other last burning questions? Everybody's dead silent now. Well, I just wanted to reiterate to everyone, and yeah, can all the speakers please put on their cameras? I think we need a round of applause for all the speakers. I see there Dave's got his camera on. Divadine, Tristan, Lizanne there, you can see them all. Divadine, have you got your camera on again there? Big round of applause, everyone, to all our speakers. Thank you so much. That was really amazing. And I do just want to sort of wrap up and say that this is just a taste of the recovery work going on in South Africa. I think you guys all realize that there is a huge amount of work on this space of species threat status and then recovery programs. And so please do check out the NBA website. We did try and link through to a lot of the recovery programs. Divadine did a great job of trying to link through to all the partners that are working on recoveries through each of the species pages on the NBA website. And then just a reminder, like iNaturalist, I don't think people really actually understand how much SANBI and our partners use iNaturalist data. So please, if you're not yet a member of or not yet a person that's on iNaturalist and posting species observations, that is the place that you can help at your own home and in your own local places. So please do look at iNaturalist. And I also just wanted to make a mention, there was a couple of questions about genetic stuff and things like that. And I just wanted to point out that from this NBA, actually, South Africa is one of the first countries to look at the new global genetic indicators for biodiversity. So it might be very interesting for you to go and read that page on genetic indicators. Yes, do we have one question? Very, very quickly then, because I'm about to wrap up.
Okay,
sorry, I can't see your actual name. Yes.
Anuri. I'm Anuri. This is about the genetic stuff, and I asked Lizanne a question about it. I'm coming from a plant background here, specifically in the Cape, where there's high levels of endemism. So I'm specifically curious with mammals. I understand, I think what Lizanne is saying is that you would try to avoid introducing related individuals so that there's no inbreeding, which of course makes a lot of sense. But what I'm curious about is in the plant world, especially in the Cape, we try not to move endemic genes from populations around without knowing how related they are. I'm just curious if that is something that's relevant in the mammal world as well, or if that's not really relevant in that space.
Yeah, thanks. That's interesting. You're talking about the sort of so-called Frankenflora that Tony Rebello always used to talk about. Lizanne, you've got 10 seconds to answer.
Luckily, my answer is not going to be very long because I'm actually not an expert. I do know that it's an issue, but I don't have a genetics background, so I can't tell you too much more. But you're welcome to contact me, and I can try and put you in touch with some of the people working in the mammal genetics space.
Yeah.
Okay,
I
thank
think
you.
this
Yeah,
is
it's more just curiosity, really.
super.
Thanks.
I think this world of genetic diversity is definitely expanding all of our horizons, and we are trying our best as South Africa to move forward in that research space. All right, I'm going to wrap up. It is 1 hour. It is 2 o'clock precisely. We will post this on the SANBI YouTube channel, so if anyone or any of your colleagues missed it, please tell them it will be there soon. And then don't forget about, we've got 2 other webinars scheduled and then a whole series coming on the NBA 2025 findings. The 2 that are scheduled already are next Friday, the 13th at lunchtime again, will be the similar series, Reverse the Red, links with freshwater species, so freshwater fishes case studies, and then like marine birds and other case studies in that nature of aquatic species. And then we do have a webinar scheduled with the Botanical Society for Thursday, the 26th of February. So that will be in the evening. And that it's a whole hour, so it's on the NBA findings in general, but we'll focus a little bit more on the plants and the terrestrial ecosystems for the Botanical Society members. So please keep an eye on SANBI socials. And yeah, see you at the next webinar. Thanks, everyone. Bye.
Bye.